Hi everyone, welcome back to Magic School. It has been quite a while I posted a video, but recently in school I had a seminar presentation, or more like a presentation for a group that we're divided in groups, and I presented for my group. So my group presented about control of enzyme level, and this is more like a mechanism or regulation of enzymes, regulation of more like regulation of metabolic pathways and regulation of these enzymes in these metabolic pathways. So now my group was to talk about control of enzyme level, and I did present about it. So yeah, let's get into it. So now, what is control of enzyme levels? Control of enzyme levels deals with the reduction or increase in the amount of an enzyme present, more like the concentration, not the activity, but the amount present at a particular point in time, depending on the condition in which the body is under. Now, these enzymes could either be upregulated or downregulated, which means the amount is either being increased or being decreased, depending on the condition of the body. Now, how is this amount actually regulated? How is the amount actually upregulated or downregulated? Now, the first one is by induction or repression of genes. Now, genes are in some videos ago or some videos before, I said something about proteins are expressed genes. So proteins are being synthesized from genes, and enzymes are proteins. So enzymes are synthesized as proteins, and proteins are expressed genes. So now the second one is hormonal control. Now hormones can exert their effects on the DNA to either induce or repress transcription that leads eventually to the formation of the mRNA, and then translation that leads eventually to the formation of the protein. Enzymes are proteins again. So now, and then we also have diets, depending on the kind of diet that is being taken in. Now it could be a starchy diet that contains a lot of carbohydrates, or contains a lot of sugar, a lot of glucose and all that, or it could be a protein, more like a proteinous diet that contains a lot of amino acids, depending on what the person is eating, or it could be that the person has a lot of fats, a lot of fatty foods, or something like that. And that is mechanism of control of this enzyme level is actually enzyme degradation. Now enzymes are catalysts, or are like catalysts, but they are not used up after the reaction. They still remain after catalyzing the reaction. They were synthesized to catalyze. So now we'll be looking at some enzymes in various pathways, and looking at some enzymes in a pathway and see how they're actually being upregulated or downregulated. Now the first enzyme we'll be considering is arginase, which is more like in the last step of the urea cycle. Now arginase is upregulated by the hormone glucocorticoid. Now how this happens is that glucocorticoid, when it is released, probably there was a high protein diet. There are a lot of amino acids, which means there are a lot of ammonia that needs to be converted to urea to be excreted from the body, because ammonia is toxic and urea is a less toxic form of ammonia. So now they need to be converted to this product, urea. So now arginase needs to be upregulated. So now the hormone glucocorticoid is released or is secreted, and now the hormone glucocorticoid is released and it diffuses across the cell membrane, because it's a steroid hormone, a lipid hormone, so it's able to diffuse through the phospholipid cell membrane. Now when it gets to the cytoplasm of the cell, it binds to its receptor. Now when it binds to its receptor, the receptor undergoes a kind of modification. Now the receptor present in the cytosol or the cytoplasm doesn't diffuse into the nucleus. This is because it's bound to a heat shock protein 90, which prevents more like its diffusion or its movement into the nucleus. Now upon binding to this hormone, the heat shock protein is released. Now after the conformation or the modification has happened, the receptor together with the hormone diffuse into the nucleus and binds to the arginase promoter region on the DNA. Now when it binds to this, it recruits co-activators and translation begins, and then mRNA of arginase is synthesized, and then it eventually leaves the nucleus to go to the ribosomes, and then the enzyme is actually being formed. Now that is how the enzyme arginase is upregulated. Now I'll be talking about another person's part. Somebody else presented with me. His name is James Isaac. He presented with me, and I'll be talking about his part also. Now he gave an example of an enzyme present in the same pathway, in the same urea cycle. But here the name of the enzyme was carbamoyl phosphate synthetase 1. Now this enzyme can either be upregulated or downregulated. Now depending on the diet that is taken in depending on the diet that's actually being taken in. So now we talked about the enzyme CPS1. I'll just be referring to it as CPS1 because that's another name for the enzyme. So now the enzyme, when there's a lot of protein in the body, there's high amount of protein diet that has been taken in. A lot of ammonia is now present. A lot of ammonia is now present that needs to be excreted as urea. So now this an hormone is released, glucagon. An enzyme glucagon is released. An enzyme glucagon is released by the pancreas. Now this enzyme activates protein kinase A. Now protein kinase A goes on to phosphorylate CMP response element binding protein, and then it becomes activated. Now this then goes on to the nucleus to bind to the CMP response element in the CPS1 promoter region in the DNA or on the DNA, and then it activates or then it then recruits co-activators that help in the transcription process. mRNA of CPS1 is synthesized, and then it goes to the ribosome, and then it gets the CPS1 enzyme. It can also be downregulated. Now when for example high amount of glucose was taken in, more like a starchy diet, now the enzyme insulin is what is being secreted, is what is being released into the body system. Now this enzyme activates protein kinase B. Now protein kinase B goes on to phosphorylate CREB, which is the same thing as CMP response elements binding protein. Now it inactivates it. Now this inactivation thereby prevents it from binding to CMP response elements, and therefore the enzyme CPS1 is not being synthesized anymore. So lastly, talking about enzyme degradation. Now enzymes, after they have performed the activity, they are left behind after the reaction. Now in a case where there was a diet of containing a lot of amino acid, a proteinous diet, now the enzymes were synthesized, were upregulated to handle this high amount of proteins that were just taken in. Now the body has passed through that phase, through that condition, and now more like a high starchy diet is being taken in. Now there's not a lot of amino acids, or there's not a lot of ammonia that needs to be converted to urea anymore. But these enzymes are still present in the cell, still present in the body. So now they need to be reduced. The amounts need to be down-regulated so the body can maintain homeostasis, to maintain a constant internal environment. So now these enzymes could then be ubiquinated, more like they are marked for degradation. Then the lysosome, a cell organ, goes on to degrade these enzymes and then down-regulate them, reduce the amounts present. Now there are other pathways where you can see this control of enzyme levels happening. For example, you can see them in the enzyme tyrosine hydroxylase, which is seen in the dopamine, epinephrine, norepinephrine pathway. We can also see it in the pentose phosphate pathway, the hexose monophosphate shunt pathway. There's an enzyme there. We can also see it in the in gluconeogenesis pathway. These are pathways that are actually regulated by control of enzyme levels, and there are probably more of them. Now in the details below this video, I'll be including these pathways and the enzymes that are actually regulated by control of enzyme levels. So that is it for my presentation. So what do you think about my presentation? Do you think it was actually enough what I presented for the lecturer? Do you think it was actually enough? Don't forget to like and subscribe. Thank you.